Greetings to all of you. Through this video lecture, I will be teaching you about metabolism of proteins in the human body. If we look into this content related to protein metabolism, then I have divided the whole content of the protein metabolism into four parts: 1, 2, 3 and 4. In the first part, I have taken the introduction of the proteins, the digestion of proteins, and the absorption of digested proteins. This will be tackled in the first part of the video. The second part, that is the second video, I will be teaching you about the amino acid pools, the catabolism of amino acids, the formation of ammonia and the transport of ammonia. This is all about, I will be teaching you in video number 2. This is the video number 2. And in the third video, I will be taking up you about urea cycle, and the fourth one we will be taking about the metabolism of individual amino acid and the related diseases concerned with their metabolism. So in this video, for the sake of ease, I am taking the most important one, that is the second part of the protein metabolism. It is all about the catabolism of amino acids, including amino acid pool. All right, so if you look into this flow chart, we eat food and we eat proteins. When the proteins are digested, these proteins are digested into amino acids. These amino acids float in the blood and reach to the different parts of the body, cells, muscles, every part they reach, and then these amino acids are further metabolized. So the total number of amino acids found in our body constitute the amino acid pools. So if we talk about the catabolism of amino acids, what happens to these amino acids for the metabolism? So these amino acids are broken down. That means these amino acids undergo catabolism. During catabolism, the amino group of these amino acids is removed out, and this step is called as transamination or deamination. And then finally there is formation of ammonia and some part of the carbon which act as remaining carbon skeleton. So these amino acids are catabolized into ammonia and remaining carbon skeleton. Now ammonia will be taking you, I will be talking you about ammonia, what happens to ammonia, where it goes, what are the different forms this ammonia is being converted in through this video. But about the remaining carbon skeleton of these amino acids, it is converted into different products like pyruvate, acetyl coenzyme A, acetoacetyl coenzyme A, alpha ketoglyceric acid, and succinic acid, fumaric acid, oxaloacetic acid, etc. These are the different substances, compounds formed by the catabolism of amino acids. And these substances or these compounds are further oxidized into CO2 and water through the citric acid cycle. So that means when we start with amino acid catabolism, so what is the main product which is formed here? It is ammonia, which is excreted out later on, we will see that. And then the remaining carbon skeleton of these amino acids is oxidized finally through citric acid cycle into CO2 and water. So this is the fate of the catabolism of amino acids. So if you look at this chart, then this will show what happens to these substances. So there are different amino acids like leucine, lysine, phenylalanine, tyrosine, tryptophan. So some of the amino acids I have written with blue color, while rest of the amino acids and substances I have written with red color. So the blue one are ketogenic amino acids. They forms acetyl coenzyme A, and then this acetyl coenzyme A enters into the citric acid cycle. While these red, the amino acids which are marked with red, they are glucogenic amino acids. That means they enter directly into the different products of the citric acid cycle, and these amino acids are further catabolized into CO2 and water. So this is the fate of the catabolism of the different amino acids. But in this video, I will be mainly concerned with ammonia. Now, what happens to ammonia? This ammonia is transported to the blood, and there it get converted into glutamine and alanine. These are the two important transportable forms. and these two substances, when reach to the liver, they again give rise to ammonia. This ammonia via urea cycle forms urea, and which is excreted out through urine. So this is the fate of ammonia. So when we start with amino acids, they are the products of the protein digestion. These amino acids, after a removal of amino group by transamination or deamination, get converted into ammonia. This ammonia is transported in the blood, go to the liver, and then through urea cycle get converted into urea, and that's why we are ureotelic. And the remaining part of the amino acids, which form the mainly carbon skeleton, is converted into these different products which enter into the citric acid cycle, and through the citric acid cycle they are oxidized finally into CO2 and water. So let us see what happens to this ammonia. So when we talk of ammonia, which is a byproduct of amino acid breakdown or catabolism of amino acids, so first of all we have to deal with formation of ammonia, how this ammonia is formed. So ammonia is formed from the amino acids only by two processes called as transamination and deamination. So first of all the transamination occurs, and then the product of the transamination undergoes deamination to form ammonia finally. So these two reactions occurs in chains, that is transamination as well as deamination. So when we speak of transamination, it is transfer of amino group, that is the transfer of NH3 plus group, amino group, from amino acid to a keto acid. So that means when amino group is transferred from one amino acid to another keto acid, a new amino acid and a new keto acid is formed. So in this example, we will see that there is an amino acid which have got amino group. It is transferred from here to the keto acid, and that's why a new keto acid is formed. And this amino group, when transferred to the next keto acid, a new amino acid is formed. So a new keto acid and a new amino acid is like this form. For example, if we start with aniline, which is an amino acid, its amino group when transferred to alpha ketoglutarate, then the aniline change into pyruvate and the alpha ketoglutarate change into glutamate. Okay, so say for example, another example is aspartate. Its amino acid when amino group when transfers to alpha ketoglutarate, the alpha ketoglutarate change into glutamate, and this aspartate again change into oxaloacetate. So these conversions are actually examples of transamination. So in the transamination, the final product we are watching it is glutamate. All right. So these are the transamination reaction. Now it is very important to notice here that three of the amino acids, that is lysine, threonine, and proline, they do not undergo transamination reaction because of their structural complexity. Now this transamination process results in synthesis of a most common amino acid, that is glutamate. Now this glutamate, which has the amino group, act as amino group donor, and then different uh, pathways are followed, biosynthetic pathways are start from here. Now this transamination reaction are responsible reversible, that means uh, if right direction occurs, then left direction can also occur. This amino group can be transferred from glutamine to oxaloacetate, and then the reverse reaction may take place. So they are reversible. We call these transamination reaction as reversible reaction. So the main component for transamination is the transfer of amino group from amino acid to keto acid, and the end result is ultimately glutamate is formed. Now this glutamate finally undergoes another step, that is deamination process. Now let's see what happens in deamination. During deamination reaction, ammonia is finally formed from glutamate. Now deamination can take place oxidatively, or it can be non-oxidative. That means if hydrogen is removed from certain substance, oxidative reaction take place. No concern with oxygen over here. So if when we say this is, we start with oxidative deamination, the first one, oxidative deamination. In this, the L-glutamate, the product of the transamination, This is formed through transamination is transported to liver for deamination. That means the product of transamination go to the liver so that they can be deaminated. So this glutamate is deaminated with the help of glutamate dehydrogenase enzyme, and here the hydrogen is removed, which is accepted by NAD, which get converted into NADH2. That's why we calling it as oxidative. So with the help of this enzyme, a alpha ketoglutarate is formed, and this again give rise to ammonia. This is excreted out as urea. Finally, we will see through urea cycle. So what is the oxidative deamination? In oxidative deamination, with the help of glutamate dehydrogenase, the glutamate is converted into ketoglutarate as well as ammonia. So this is the final formation of ammonia through deamination process, oxidative deamination. Now this enzyme is allosterically inhibited by GTP, ATP and NADH2. And medicinally, why this enzyme is important? This enzyme increases in liver disease in the serum, and when we examine this enzyme in serum in the liver diseases, certain liver diseases, then this enzyme will be at higher concentration, that is glutamate dehydrogenase. Now this reaction, oxidative deamination, it is catalyzed in mitochondrial matrix of the hepatocyte, that is it is taking place in the liver cell, but sometimes in some other parts of the body this deamination also takes place, like muscles or brain, etc. When we say non-oxidative deamination, then the enzyme is serine dehydrogenase. So for example, amino acids like serine and threonine, they are directly deaminated without any involvement of removal of hydrogen, and a pyruvate and ammonia is formed. In both the cases, we see that the ammonia is formed. So deamination is a final step for the formation of ammonia in the liver. Now what happens to this ammonia which is finally formed in the liver, or it is formed in the muscles or brain? So all the ammonia has to be accumulated in the liver, and from the liver this ammonia will be converted into urea. Now ammonia is highly toxic. We say the ammonia is very highly toxic. It cannot remain in the body for a long time, otherwise it will cause disturbance in the body. So it is transported from one part of the body like muscles to the liver via blood in non-toxic forms. So ammonia formed in extrahepatic tissue like brain, muscles, etc. They are transported as non-toxic metabolite to liver and kidney, from where they are excreted out. Now ammonia is mainly transported in two forms, that is glutamine as well as alanine. Ammonia is transported in these two forms. So first of all, transport of ammonia as glutamine. So glutamate combines with ammonia. With the help of ATP, it get converted into ADP and Mg plus 2 plus ions. Glutamine synthetase is the enzyme. So this reaction results in the formation of glutamine. So that means that ammonia is incorporated in the structure of glutamine. This glutamine is non-toxic, which is transported to liver via blood. This glutamine along with water and glutaminase, again inside the liver, in the liver it is broken down into glutamate plus ammonia. This ammonia which is formed in the liver is converted to urea via urea cycle. That is another cycle. We will not be discussing that urea cycle here. Alright. So this is the form, that is transport of ammonia as glutamine. Now another form of transportation of ammonia, it is as like aniline. The transport of ammonia as aniline, it also takes place. So aniline is non-toxic form used in transporting ammonia through glucose aniline cycle. So there is a cycle called as glucose aniline cycle, and through this cycle the ammonia is converted into aniline. Okay, look at this diagram over here if you are visible. So I have shown you the muscles and I have shown you the liver. What happens in the muscles? The muscle proteins get metabolized into amino acids. These amino acids release NH ammonia. This ammonia get converted into glutamate, and the glutamate then supplies this NH4 to pyruvate. Alright. So in this reaction, when the glutamate gives NH4, that is ammonia, to pyruvate, which is present in the blood byproduct of glycolysis, the glutamate change into alpha ketoglutarate, and this pyruvate get converted into aniline. So in the muscles or brain, this is the reaction which takes place for the formation of aniline. That means ammonia, which is a byproduct of the amino acid breakdown, first of all incorporate itself in the glutamate, and then from glutamate it is transferred to pyruvate, and the pyruvate get converted into aniline. So this aniline is non-toxic form which is transported through blood and then finally reach to the liver. Now let's see what happens in the liver. Inside the liver, the aniline gives amino group NH4 to alpha ketoglutarate again, and this alpha ketoglutarate again give rise to glutamate. So that is, glutamate cannot be transported, it is transported as aniline. The glutamate finally, with the help of glutamine dehydrogenase, forms ammonia, and this ammonia inside the liver is converted into urea via urea cycle. So this was glucose aniline cycle. That means, first of all, in the muscles or brain, I have shown you here in the case of muscles, the aniline is synthesized by this pathway, and then aniline is transported to liver, and then aniline again get converted into pyruvate, and finally glutamate is formed. All right. So this is the way this aniline is transported. So this is the write up of what I have shown you through the diagram. Inside the muscles, amino acids are forming glutamate, and in the liver, the glutamate again give rise to this ammonia, which is converted into urea via urea cycle. So this is how the catabolism of amino acids during the metabolism of protein takes place. I hope this video is a fruitful video for you. Thank you very much.